How to make a bacterial glycerol stock. A bacterial glycerol stock is the suspension of bacterial culture in glycerol. Also known as free stocks, glycerol stocks are an effective way to store plasmids for later use, ideally for an indefinite period of time. Glycerol is a cryoprotectant. It stabilizes the frozen bacteria and prevents damage to the cell membrane, ensuring cell viability by preventing ice crystal formation during freezing and thawing. Additionally, it is non-toxic and compatible with a wide range of bacterial species. Lastly, it is cost-effective and convenient. In plant transformation, glycerol stocks are routinely made of both E. coli and Agrobacterium. After going through the time-consuming and costly process of creating a plasmid for plant transformation, you'll want to store the plasmid for later use. On a bacterial plate, those are stable at 4 degrees Celsius for a few weeks. While storing plasmid DNA is convenient, there's not an easy way to create more of that DNA when you need it. Bacterial glycerol stocks allow for stable storage for years, and it's easy to get more DNA when you need it. The materials required for making a bacterial glycerol stock are the plate with your colony of interest, liquid LB media, the appropriate antibiotics for your plasmid, bacterial tubes, 2 milliliter cryogenic vials, a tube rack, 80% glycerol, sterile 10 and 1000 microliter pipette tips, and a heated shaking table.
Right now, I'll be demonstrating day 1 of making a bacterial glycerol stock. First, you're going to take your labeled bacteria tube and add about 3 milliliters of liquid LB media with the appropriate antibiotic added. In this case, the appropriate antibiotic is ampicillin. Next, now that your bacterial tube has the liquid media in it, you're going to want to inoculate it with your bacterial culture. Using an unfiltered P10 pipette tip, I'm going to very gently tap onto my selected colony, which you can see is circled below. Just gently tapping the surface will do. You don't need to worry about getting the whole colony on the tip. Then eject the pipette tip into your liquid media. After that, tap the tube, and now you're ready to go grow your bacterial culture overnight at 37 degrees Celsius. After you've labeled your cryogenic vial with your plasmid information, which is the name of the plasmid, the antibiotic resistance, the date, and your initials, a pro tip is to take a small piece of just regular office supply tape and apply it over the label that you've created for your cryotube. Now, when you take it in and out of the freezer, your writing is less likely to rub off. Add 300 microliters of 80% glycerol. Add 300 microliters of 80% glycerol. After you've added the glycerol to the tube, you can now add 900 microliters of your bacteria that you grew overnight. Tap your tube and vortex briefly. Now that you've suspended your bacteria, you're now ready to put it inside the freezer.
Now it's time to document our freeze stock. Our lab uses an Excel sheet that is stored on the cloud and backed up regularly to track the location and contents of each tube in the minus 80 degrees Celsius freezer. We record what box it's in, the slot it's in, and then information about the strain, such as the resistance, a reference, the genotype of the cells, a description of the plasmid, as well as the owner and the date it was added to the freezer. In addition to the documentation in the Excel sheet, it's also a good idea to include a plasmid map. Our lab recommends adding a thorough description to the plasmid files and note if this plasmid has been sequenced, as well as annotating any important features.